Well, hello and welcome to Bio 211. We are looking at Chapter 12 in Anatomy and Physiology 1. And what we're doing is we've worked our way up to how do we use graded potentials to activate action potentials, and in this case, using a multipolar neuron. And in the previous presentation, in blue, what I demonstrated is for us to actually activate an action potential, we had to have some form of signaling that arose at the dendrites in cell body that allowed for a series of stimuli that are depolarizing, that get to a specific value, which is minus 55 millivolts, that then cause the opening of voltage-gated sodium channels. And in that voltage-gated sodium channel opening, we depolarize the actual membrane, so it gets to a plus 30 value. The consequence to that is, simultaneously at reaching plus 30, we now have opened the voltage-gated potassium channels. You'll see in this little part right here. And these stay open until we have hyperpolarized the membrane. And what's happening is you actually have an absolute refractory period and a relative refractory period, where in the relative refractory period, if we had extra stimuli to push this up to minus 55 quickly, you could activate another depolarization. Some things that seem to confuse students. Okay, all of this here is now occurring in the green part. Right? All of that, and it's going down, down, down. Because this is myelinated, this is going to use a specific type of conduction, and that conduction is saltatory conduction. So it's going to travel really quickly. I want to make sure you get that, number one. So this isn't just happening once. It's happening millions of times to actually travel across the entire surface, ultimately causing the release of neurotransmitter in a regulated manner that requires a voltage-gated channel opening here at the axon terminal. Okay? So with that in mind, let's think about what's actually happening at the individual channel levels. And what I mean by that is the pair of channels that are found adjacent to one another along the entire axon and axon terminal, where you can see here is a sodium channel, here is a potassium channel. These are both voltage-gated sodium and voltage-gated potassium channels. And that's what you're supposed to see down here in these tracings. The new information is that the sodium channel has two parts to it. It has an activation gate and an inactivation gate. And the potassium channel has this occlusion mechanism, right? So when this is at rest, minus 70, the activation gate is closed. The inactivation gate is open. When we depolarize it, notice the activation gate is now open. So now sodium can move in. Yay! You get the idea. At a specific value, plus 30, the inactivation gate closes. Whang! No more sodium in. Now, potassium can exit, and it stays exiting until such times as we approach our repolarization, right? Meaning the hyperpolarization phase. And notice what happens here. The voltage-gated sodium channels are now shifting who is open and who's closed. That's the tricky part. If there's anything that's tricky about this, is it has to return as quickly as possible to the original conformation or the original shape of those gates being open and closed. Because you're going to see here that this is closed at rest, but it's open now when we actually get to plus 30. Okay? Cool. That took too much time to explain, but it's really important. And notice I'm paying attention to which gates are opening and when, and I'm tying it directly to a voltage charge. Okay? That can be measured. Cool. So having done that, let's go talk about what's actually happening here. Okay? Because we have two types of transfer. And you'll notice in the previous slide I mentioned, oh look, this one's myelinated, so it can travel faster. I want to make sure you understand that there are two types of conductions that can happen on an axon. Because now in green, according to your textbook, right? In green, we are going to have, that's why they have the green there, right? Voltage-gated channels using action potentials that are traveling along here, right? Okay, so in continuous conduction, there is no myelination, and therefore it goes from channel to channel to channel to channel to channel the whole way through. So sodium, potassium, sodium, potassium, sodium, potassium, sodium, potassium, the whole way through. The way it works with saltatory conduction is by having a really large diameter, okay, or even a medium-sized diameter, and myelination. You can concentrate the actual channels in between these spaces. And what ends up happening, the way your textbook describes it, it says it leaps. Don't ever tell me it leaps. But essentially, the charge travels more efficiently across the surface of the membrane because you have these really large numbers between the nodes, okay? And now what happens is it can travel more quickly. Wham, 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 as opposed to dun, 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 dun. Okay? So that's why saltatory conduction is engineered. And from your reading, you know that there are A-type fibers. So I'm just going to put A, B, and C. Okay? And this is an internal quiz inside inside my presentation. What are A-type fibers? Can you answer that? What are B-type fibers? Can you answer that? And what are C-type fibers? Let me give you a hint. A-type fibers, those were our sensory afferent neurons. C-type fibers, those were our interneurons. Okay, cool. So what are we going to do? We're going to send this information all the way down to that synapse. So let's take a look at the synapse inside of a cell. Okay, I remember at the beginning I told you there are actually two types of synapses inside the body. The first of which is an electrical one. We saw that back in chapter 4, where you actually have ion current spread through gap junctions. Okay, and it's faster, it's two-way, you can actually synchronize groups of neurons. I'll take that out of there. Okay, groups of cells, just put that there, cells. Because we only teach it to you in terms of how it works for, say, cardiac muscle fibers and stuff like that, and smooth muscle and osteocytes. Okay, but there is chemical. Okay, and chemical means we're going to release a neurotransmitter, but that also means that you can actually have different synapses that form. And inside class, I said, look, don't pay attention to this all that much, and that's really true. But Mother Nature needs to regulate those chemicals so that even if a neuron has depolarized, right, and it's become an action potential, you can still influence it. But that's just more FYI than it is anything else. So what does it look like? Well, here is our axon terminal with our synaptic end bulb, and we have now decorated at the end. So let's draw a multipolar neuron for you so you know what you're looking at here. Yay. If you're a student who's like, I don't need that drawn for me. Well, good, fantastic. That's the cell body. Here's an axon. Here are the axon terminals. Yay. This right here is what you're looking at right there. Okay, so I'll put nucleus there. Yay. And if you're bored, figure out what the different parts are and explain them to me. Okay? Imagine you're teaching it to me. So that charge is coming along here, right? That is a depolarization, voltage-gated sodium channels opening and closing, voltage-gated potassium channels opening and closing, till we finally open up our voltage-gated calcium channel, which means now calcium moves in and it binds to our vesicle with neurotransmitter inside of it. Okay? And inside our vesicle with neurotransmitter, now because calcium has bound to it, it can fuse, and now exocytosis occurs, and that's going to bind now the ligand-gated channel on the opposite neuron, which is now going to be called the postsynaptic neuron. Postsynaptic neuron, because up here now we've named this our presynaptic neuron. And this is fundamentally different for us in terms of our way of thinking, because these are a chemical synapse with a synaptic cleft that uses a neurotransmitter to actually then activate. Let's just say this is sodium, so therefore this is going to be now a depolarization on that membrane that's going to travel to the axon hillock. And if you have sufficient neurotransmitter, you'll end up causing now an action potential, right? Yay. That will then travel along the entire axon. Okay? Fantastic. At least I hope it's fantastic. Let's get rid of that. Move on. So what actually is going to happen here? Well, what you want to understand is that not every potential is excitatory. That's just the easiest way for us to teach it to you, right? You can also have inhibitory postsynaptic potentials. And what's significant here is that inhibitory means that you'll probably let these guys in. And normally I teach it so it's chloride ions. When I start using potassium ions, students get really confused. But the consequence is still the same, right? If we're letting a negatively charged ion in, it makes sense that you'll have a hyperpolarization. And that hyperpolarization will stop all signaling at the exon hillock because it will never get to -55. So how does this actually work? Well, when we're looking here, we can have different types of receptors on the surface of those cells that will allow us to modify what happens at the membrane. And in this example here, you have an ionotropic acetylcholine receptor where acetylcholine is the ligand. It binds the acetylcholine receptor, and now the ion travels in. And in this cartoon, they have multiple ions traveling in, which is going to confuse the snot out of everybody in the room. Okay? But the idea, of course, is you've now caused an EPSP. In this case, we are going to use GABA, gamma-aminobutyric acid, and what's going to happen is it binds this structure, okay, and now chloride ions are allowed in. And that chloride ion, of course, is negatively charged, and now you've caused an IPSP, okay? And that looks, whoops, let's go like that. Let's go like this. To make sure you understand what I mean, a graded potential as a result of this would look like that. A graded potential as a result of this gamma-aminobutyric acid letting chloride in does this. Metatropic receptors. So somebody mentioned in class, no, in AMP2, you're going to see this thing that's called a G protein coupled receptor, right? That's what they're talking about here. You have a neurotransmitter binding a specific receptor that then uses intracellular signaling to then open up that channel. And this is an example of, in this case, acetylcholine using G protein to now allow, notice which way the ions are moving. Potassium ions are moving out, and that causes an IPSP. And that's why students get really confused. Okay, so that causes that. Okay, cool. So now we have all these neurotransmitters to start thinking about. And how do you actually get rid of them? Because if you leave them hanging around, you can see how you could cause all sorts of depolarizations and hyperpolarizations, and that could be really confusing for a cell. So instead, what Mother Nature does, let's get rid of the cartoons. What she does, she gets rid of them. She'll allow them to diffuse against their, or down their concentration gradient. And the significance here is going to be that astrocytes will degrade them. Okay? Simultaneously, cells will make enzymes. The one your textbook introduces first is acetylcholinesterase, and I'll put this like this. ACH. Notice I'm putting a capital E here for you to understand that we can break down acetylcholine with that. There are two more that I put here. Catecholamine O-methyltransferase and monoamine oxidase. You'll see when you're reading the significance of these. And then you can also uptake or reuptake would be the way to put it. Okay. By neurons or glial cells. And the glial cell, of course, up here would be the astrocyte. And you can actually break it down there. Okay. Makes sense? So Mother Nature can't have neurotransmitters kicking around for a long time, meaning inside spaces where they could randomly bind to receptors. And what this means, of course, is that all neurotransmitters should cause some form of summation. Okay, so that's where we're going to go with this. If several presynaptic M bulbs release neurotransmitter at about the same time, it should be summed, and there are two different types of summation. Okay. You could either spatial or temporal, and your textbook gives you an example of this. So this is going to be temporal. Temporal is here, where you have one neuron releasing increasing amounts of neurotransmitter to then cause the depolarization at -55. Okay, so that would be at the exon hillock. But over here, if you've got multiple neurons releasing smaller amounts of neurotransmitter, you can actually get to -55 more quickly. Okay. So this is time in milliseconds. That's how you can tell what's actually going on here. This is a good opportunity for us to stop here, and then we're going to introduce the concept of neurotransmitters in the, I believe, the last presentation. Okay?